So the first prompt for this breakout room is, wouldn't it be great if, and then fill in the blank.
So maybe I can start with a question. I'm not more interested in bio side, not really. So it's kind of fundamental question about aging. I know this is a question that came up a lot. Is there an agreement or some hypothesis model on how do we define health span? Like when does a cell go from like aging, like opioid versus that makes sense.
Oh, yeah, that's a good question.
Like, are there
No.
any markers? So is there?
Oh,
any-
well, I guess there could be some markers. I think one of the challenges is, so a lot of the biology of aging just goes back only 30 years when some of the first mutations were discovered that it's stem-like. They were found in C. elegans. And then, so you have a mutation that's positive of that. So that made people think, oh, there's a genetic basis that was sort of accepted. And so I feel like we're still in the heavy discovery of things like that, like what are the coordinates of what would something look like? So I think that there's two horizons for synthetic biology. This is a long way of answering your question in the context of what we're doing. First, I think synthetic biology could potentially provide tools for interrogating aging systems and cells and everything in a more systematic, rigorous way. And that could be through the cycle that, I don't know, the 4 talks already had, but you got the input, you got the manipulation, and then you got the outputs. I think we need all of those still to be done better. So that we can answer your question and say, oh, this is the thing. When this changes, then all of a sudden things happen. Just
So
one.
one answer to this prompt in terms of knowledge would be identifying aging markers.
Yeah, that's right. Yeah. Like what are the markers? What systems are driving aging? In
Hi,
particular?
sorry, this is Jessica online. Could everyone make sure to speak into the microphone so that we can all hear online and through the recordings? Thank you very much.
Okay, there's just one central microphone, so we'll just have to speak up, I think is what we'll do. I think it's just
Yeah, you
that.
sound great now.
Yeah,
Okay.
we'll just speak in that direction.
Should we, is everyone down?
Yeah, why don't we shift? Excellent.
Thank you very much.
Is your one or my company? Just the last context thing that we didn't really go over in the introductory talks that I think is really important is that aging is not just getting old. There's a distinct change in the trajectory of decline, and it's the acceleration of that rate of decline with age, biological age, that is the hallmarks of what aging is. That's called senescence. And so if you look at most animal models, they all follow the same pattern. If you're a worm or a mouse or a human, where there's, now that we've addressed most of these infectious disease things that lead to especially early mortality in humans, you have a pretty long plateau runway of very, very low rates of mortality. And then around 60 years old, it accelerates and goes down very quickly. Your survivorship does. And so it does seem that something changes And it happens at all these systems, not simultaneously. That's where the stochastic part is, but you lose muscle mass, you start having cancers at much greater, and dementia towards the end just explodes in frequency. So I think there's a lot of independent systems that all kind of give up at the same time. And there's a lot of evolutionary theories about why that should be. But that gets to what are the hallmarks of aging, is like, you'd like to know which things are maintained and then which things start decline. And that could be, there's as many theories as there are aging biologists, epigenetic markers, it could be protein accumulation and loss of proteostatic maintenance. It could be telomeres, which used to be a hot topic. I'm not sure they're so, it seemed like an easy solution. That's why I think they got a lot of attention. And all these different sorts of things, cell communication, insulin resistance, insulin sensitivity, fat cell metabolism, all of these kinds of things seem to be not related, but the question is, are they related? So super long answer to your question. We don't know what the biomarkers are. That's actually a big push in the NIA is to try and define those sets of things. And again, so I think figuring out a way to interrogate those systems and then to create synthetic biomarkers, I think would be really interesting. Like, are there ways of creating a system that the cell transitions into some new state and then it sends out a signal saying I'm in trouble or whatever. I think there's a lot of basic biology of aging we don't know before we think about using synthetic biology to cure aging, for instance. So, but I do think that's important.
Follow-up question. So you said there's a distinct change in the trajectory and rate of decline.
Yeah.
So is it rate? So, loss of muscle mass occurs really, really slowly and then starts going more rapidly. Is it purely rate?
Yeah,
Okay.
for the most part.
We start aging from the born, ourselves start aging from.
They do, but there's something goes wrong around 6 years old that all of a sudden the rate of decline, and again, it's going to be very tissue specific because some things are proliferative, like even muscle, where exercise. You can, I don't know if you've ever seen like the world champion sprinter at the 100-year class or whatever. It's great. Like he's moving down very slowly, like he works out, but he can't build muscle mass. Like there's something about his body that it will not respond to the normal feedback signals that even a 4-year-old would be able to run just much better. And it has nothing presumably to do with, well, it's hard to know. So it's that kind of thing. Some things could be a decline in the maintenance cycle. And then some things like our brain, which are not turning over very much, presumably is due to just accumulated damage and a decline in the maintenance function of some of those activities. But I think it's quite heterogeneous depending on tissue.
From 60-year-old for human is the threshold.
Pretty much. Yeah.
That's depressing.
Yeah. Coming up
I'm
for me
getting there.
next year. I'll be 60 next year. So I've been looking at this for a long time. I'm like, oh man.
Get into the workshop.
Yeah, I know, exactly. It's not instantaneous. That's the other thing. It's very variable across different people, different animals, all that stuff. But when you look in aggregate from a kind of an epidemiological point, and then it just keeps going.
Yeah, we see that with senescence markers as well, that there's a difference in relationship in old and young age. We for instance, if you look at monocyte senescence markers in the blood and you break longitudinal studies like the BLSA into younger than 60 and older than 60, there's a slight inverse association with, say, grip strength, like markers of frailty. If you look at plasma senescence markers in people, and then if you look after 60, there's a much sharper inverse association between senescence markers and grip strength and other markers of frailty. It's pretty interesting.
So what are these markers, and are they causal or are they?
It's a good question. Could be both. There's clearly a lot of factors going on.
And so at the cellular level, My understanding is also that it's not entirely clear the causative versus correlation, right? Like it's not necessarily telomere shortened.
Yeah.
That's
Yeah,
It's like all these mark, like the wheel is a wheel because it's not quite clear what the causative correlations are.
that's right.
Correct.
That's right. So I think proteostasis, I think it'll be interesting as these things go, is like the synthetic biology. Should we just be thinking about cellular biology as a primary driver of action versus kind of the tissue interactions and those kinds of things? Because that might create different kinds of things, but so proteostasis seems to be an important thing in aging. Get accumulation of damaged proteins at some point. Just by chance, you're going to get some protein folds in some weird way that the proteasome can't degrade, and then it becomes sticky, and then other proteins stick to it. And so there's this arrow of time aspect of things where some things you can't back out of, you can't fix, and that's one theory of cellular aging is the use to accumulate damaged proteins. And then in yeast, they try and package them into the
Right.
old cell, and then the new cell starts with undamaged proteins. And for a single cell organism, that's one solving that issue. But for multicellular organisms, that's where these these cells that are hanging around after they're dead, they're not really dead, but they're accumulated a bunch of junk.
So another question actually I have about this is that that how can we implement synthetic biology to slow animal aging? I can understand how to use synthetic biology to understand aging biology, right? I mean, to help us repair the network or whatever, but how to implement it to slow animal aging or even human aging? I mean, the genetic circuit, we just do gene therapy for everyone. Or I just so yeah, we have this recent oscillator paper, and the question I have always is how can this help human? And I cannot say that we just inject this circuits into you and then it will kill everything. I mean, how to do that? I just don't understand how synthetic biology can be implemented to slow aging. I can understand, I mean, CAR T is cell-based therapy. This I can understand, but for aging, should we develop cell-based therapy for aging? Or I don't think people want to try gene therapy for aging, right?
I have some thoughts, but I don't want to dominate. Maybe in your space, what other people think.
Go for it.
Well, so I think your concerns are about delivery, right?
Yeah.
And I agree with you. I don't think we'll be engineering humans to, turn on stress responses
Mhm.
in a transgenerational way. Like that's just ethically complexity. There's lots of reasons we won't do that. I think the question is, are there other delivery modalities that could be used?
That's right.
So Josh and I were just quickly talking about in the first break was, so I think that the biggest challenge is going to be the brain, right? For many reasons. But could you engineer a cell as the therapeutic that you would inject into the brain that then send out signals or had some sort of replacement? So you're not going to be able to replace neurons because neurons are who we are, the connections among them. Right? Is there something that could be made more healthy? Or that's why I was wondering on the blood side, there's all these cancer-based therapeutics where you genetically engineer bone marrow and then you have it produce something and then put it back in. And so you can use gene therapy as a therapeutic if you keep it somatic, not germline. And that's totally fine. It's really not any different than giving someone medicine. So I think maybe there's these subclasses of things where you're not replacing whole tissues, but can you do that? And then we have, obviously, through COVID, we saw the power of RNA. So could you introduce artificial proteins into human cells so that they'll produce some RNA that is then used? I think the big challenge is in the synthetic biologists working, especially example, is how do you do a whole circuit of something? If that's what we really need to do, then that's going to be a challenge.
Okay, basic cell-based therapy too.
It seems like that would be the
Okay.
first mover, would be to engineer cells that you would then introduce into human tissues to help. So sarcopenia, I gave that example. I know nothing about muscles and the physiology of muscles, but one could imagine that you could engineer muscle cells, and then that whatever the cause of sarcopenia is, that then we're able to be more sensitive or able to, regrow, or they send out the appropriate signals, or they send out a signal to kill off the muscle cells that are blocking muscle growth, or that kind of stuff. That's a pretty accessible tissue where we know exactly what to do. And so I could well imagine using that as a therapeutic. Any of these blood-based things, if there's these famous, it's not my area at all, but there's these famous vampiric aging models where you take blood out of a young individual and then put it in an old mouse, and then all of a sudden the mouse is better. That's got to be biochemistry, right? There's not something magical that happens with young blood. There has to be compounds in that blood that are being transmitted. So if that's true, then presumably you could engineer those in bone marrow and have the blood be produced. That's doing, and I'm sure there's people working on that.
So in addition to blood cell and immune cells, other cell types that we can also inject, like fibroblasts or epithelial cells, can we inject that into human body? I don't know. I mean, I'm completely not in the field.
Yeah.
I can understand blood cell. I can understand T cell, macrophage, but I cannot understand other cell
Yeah,
types.
I don't think were there neurobiologists here? So that's a missing group of people, frankly. Because I think people are working on Parkinson's treatment to kind of inject some neurons or something like that to kind of restore the dopamine production in the parts of the brain where Parkinson's is.
Okay, interesting.
But that's another application of that I've thought of synthetic biology, is instead of saying, oh, this cell is injectable, this one's not, how do you engineer a cell to be a therapeutic in the appropriate thing? So if the big thing is we need to address frailty and muscles because we need these muscle cells, well, engineer the muscle cell to do the job that you need to do. Easy to say, I mean, this is a 100-year project probably, but that's the kind of synthetic biology. That's why I think this has got to be the total future of all this, rather than just kind of taking the biological diversity we have and saying, well, this is what we can do, or this protein exists, let's tweak around with that. Well, how do we make whole new proteins? How do we make whole new circuits? How do we make the cell do what we want it to do, rather than just saying, taking it at face value? But then we have to use synthetic biology to discover.
Yeah.
why it doesn't do it.
That's right. Yeah, that's what synthetic biologists do. I don't know how to, but that's good. So because what we are trying to do now is to engineer HSC, the blood stem cell, because of what you said. I mean, that you can transplant it back to the animal. Other than that, I don't know other cell types. I know T cell, of course, a macrophage. You can inject. Basically, you get some human cell. You engineer it. I mean, assuming that we know how to do the circuit engineering, and then we can inject back to human to help.
Well, I think also the quickest term, which is is through some sort of compounds or drugs. So can you create synthetic systems that are better readouts of drugability? So again, if we know what the health states are and could have readouts, then could we do large scale screens? So for me, the
Okay.
whole problem of
Help
the
with-
field is like, how do you create scale? That's what engineers should be good at helping, right? It's like, how do you create scale solutions to these problems? How do you assay the effects of 100,000 different compounds on, the neuronal maintenance through aging? Well, you're not going to do that in 10 million worm, or 10 million mice, right? Or you're not going to do it in any humans. So I think you're going to need to engineer some microphysiological models, some sort of, some high throughput, which is organoid, something that allow us to do that. That's going to take it in. It's a physical engineering problem, like how do you even deal with all those cells? And then there's a bioengineering problem of how do you create the readouts? How do you, again, engineer something rather than saying, oh, these neurons don't like to grow in artificial environments. Can we create a model neuron that actually does, is still useful, but is then screenable?
Sorry.
And then I think you can do genetic screens, you can do drug screens, you can screen for complex tissue interactions. There's lots of things. And to me, that's the blue sky thing that's not, just from outer space. It's just like, if we could build screenable human tissues, then and bioengineer those tissues. Again, it's not a therapeutic, then, but it leads towards therapeutics.
Yeah. That's totally right. Yeah. Well, the other thing that we may be able to engineer is the microbiota that interfere with aging, and that's tractable. Some of them are tractable, and then we can easily design synthetic circuits in some of the.
I was kind of curious on your knowledge, how much like the aging field has looked into how the microbiome affects aging.
A little, again, it's not my specific area. People are starting to look at that. I guess there was two aspects of that. One is like the natural microbiota. And then I thought what I heard in Inkling was that could you engineer the
Exactly,
microbiota
yeah.
to be like its own little drug delivery system or something internally. So I don't think we really know anything about those rules yet. Obviously, people have done this again in mice with, fat versus skinny mice and those kinds of stuff. And since we know that whole metabolic disruption and maintenance is a key part of longevity pathways and predictive pathways, it's got to play a role for sure. There might be literature has gone into that more depth than I.
Yeah, and there was a paper about microbiota effect of metformin to right. I mean to people, to mice. So also relevant.
That's right. A lot of these metabolic disruptors directly affect the microbes in your.
Yeah. That's easy to transplant. So yeah.
I'm not familiar with that paper. Was that saying that the metformin had an impact on the microbiota that would then have an impact on the person?
All the microbiota, I don't remember either. But I remember there is interaction, so it depends on the microbiota. All the metformin maybe affect the microbiota to affect. Yeah, I don't remember the details, but I remember there are interactions there. Yeah.
In our worm experiments, that's how it works. Actually, the metformin affects the bacteria. The bacteria affect the. There's some complex thing going
There's
on
like
there.
a
If you use dead bacteria, the metformin doesn't work.
Really, like not at all.
Yeah. So is a bacteria creating well, we don't know. Is it creating a metabolite of metformin that is then the active drug, or is it the metformin is changing bacterial metabolism with them that extends the life of the worms? We don't know. It's complex.
I guess another question that I had, are like most of these aging phenotypes or senescence phenotypes more related to like loss of certain functions, or is it like a mutated form of something? Like what is usually like, or is it just like a mix of everything?
I think it's everything. But that's, again, I think we need to figure out. I think in the positive effectors that we know about, say, with this insulin signaling pathways and those sorts of stuff, I think the fundamental biology is always from the organism's point of view, is should they make an investment of energy in maintaining themselves versus reproducing?
Mhm.
And so the historical reasons why we have all these systems is to make that choice. And in human society, Western human society, energy is not a problem. It's a problem because we have too much energy, right? So we could drive these systems to being much more energy consumptive. And so, like, why don't we have better vigilance of mutation? We have the proteins to check for replication integrity. Why isn't it just go crazy? And just be very careful. At some point, enough is enough, and you waste energy by doing more. Can also clog up the cell. So in most of those things, what in the animal models, it's a switchable system. It's like, do you invest in protecting the cell, or do you invest in reproducing? And so those are all positive things that probably could be turned on and maintained. In other things, it's the maintenance, which we don't totally know, like the epigenetic state of the genome. That's get laid down when the cell is differentiated. Do those markers just slowly, under biochemical attack by just what's going on in the cell? Do we just slowly kind of lose those, and then all of a sudden, in your your intestinal cell, you're turning on skin cell genes because they lost their suppressive markers, and maybe that's partially what drives aging. Obviously, just raw mutation itself, probably not as important a factor, except in replicative tissues. Yeah, so I think all of those things matter. How do you maintain? And I already talked about proteostasis, that you just started garbage in the cell, and the cell has some garbage collection systems. It's really exquisite. Again, evolved like 1 billion years ago, these systems, so they're obviously really important. They're conserved. But at some point, the garbage overwhelmed. As you go long enough, that's kind of the theory, the theme of aging is like, if you push things long enough, eventually something will go wrong, like your car, and you get a couple of proteins that stick together and they can't be degraded, and then they start getting more friends, and then at some point the cell has too much garbage protein. There's nothing it can do about it because its systems can't attack it, or you accumulate a mutation or you lose a genetic marker or something like that. All these are time-based probabilistic things. And so the solution for aging is to push those probabilities out, is one solution, or in multicellular replicative tissues is to kill the things. This is what we do with cancers, right? It's like we just want all those cells to get killed, and then to keep the healthy tissues, be the ones that. And I think that's kind of, again, the use to some degree. Do you agree with that?
Yeah.
So sort of that's another blue sky kind of thing is, are there novel, ignoring the therapeutic problem, but are there novel protective systems that could be introduced that are artificial? Oxidative scavengers or something like that, or could you take a lot of the great work in bioengineering has been to take something from one organism, let's say a really oxidative resistant organism, as this kind of protein for this job. This organism over here, this wimpy mouse or human, hasn't ever been bathing in oxidative whatever, but if we move that protein changes into this organism, all of a sudden it'd be even more resistant. So the selection hasn't led to that resistance, but you could artificially engineer that resistance in based on what we know about other systems. So that would be another way of trying to create kind of artificial aging resistant things, would be to kind of engineer novel protective system. I guess I've already talked about the sensors of health span, which you brought up straight out, because I do think that that's one of the biggest problems, is we just don't, we have no way of monitoring what's going on in the system.
Could have sentinel cells, in our tissues.
Yeah.
Kidneys. aging or aging, tissues are aging.
Yeah.
They could give some sort of readout.
SOS.
Yeah.
It could go in the
Yeah.
blood, we could just get especially if it's sent out to like a DNA signal, we could just sequence our blood periodically for the markers.
Yeah.
Would be some important capabilities of those sentinels. Can you
Sorry,
do-
say that again?
What would be some good capabilities of those sentinel cells?
Well, first diagnostic, I would say, and then eventually converting the diagnostic into some sort of therapeutic response. Yeah, that's another question then. So we talked a lot about understanding aging. How do you reverse it or delay or like what is the consensus in the field? Because we talked about, or if there is any, or what are the idea? We talked about telomerase a little bit, and I don't know if it's kind of like an old idea or what. But even if we are able to understand the aging markers, right? Say you have a readout of proteostasis somehow, which you can do, right? You could build something like that. Then what do you do?
Well, I think it depends on the tissue and the organism, but if that cell would be more aggressively committing suicide in a lot of organisms, that would be helpful.
Okay.
So that's the Senolytic model is basically you take drugs to make cells die or kill themselves if they progress to a state where they're no longer healthy and able to maintain function. Because the corollary is if they're not maintaining healthy function, then they could be sending out unhealthy metabolites. And so you would like them to just, if they would die, that would be a thing. I think manipulating the metabolic state of cells is another thing that people are trying to do. So a lot of these rapamycin, those kinds of things are shifting kind of the sensitivities to metabolites and those kinds of responses. It's kind of, I think there's innate immune protective things that are conserved within cells that start manipulating those, then cells are more protective of themselves. So those are useful targets. But for the most part, those targets, like much of medicine, are defined based on outcomes, not based on mechanism. And so you find that this makes a mouse live longer, therefore you start, and then you don't know why exactly. And so it seems like we're way far away from a rational. It'd be nice to, again, to be able to say, here's a set of 200 compounds that all work. What are properties of them that are in common? Could we then rationally design some, or which systems are they targeting? Those kinds of stuff. So it's still early days for sure for that.
So would you want, as a therapeutic solution, would you want to make the cell commit suicide, or would you want to eliminate resistance to apoptosis? Like my understanding is senescent cell resist the activation of apoptosis, right? They
Right.
sit there and then they secrete all this stuff.
Yeah, that's right.
So would you want to, because it's. I don't
You
wanna
want
say
them to
easy, but-
die. So yeah,
Right. So
whichever,
it doesn't matter
whichever
how.
way, yeah,
Okay.
whichever way you do it.
There are work on engineering like CAR T cells to kill these senescent cells. I think
Yeah.
people already did it. So,
Yeah, so that's another immunotherapy.
yeah.
And again, that could be engineered back to the therapeutic. If that was super useful, you could make, this is how zombies movies start, but you could create the,
Like a kill switch.
you could,
that
right,
is-
and then you could create a set of an engineered immune circuit that then went out and was a little more vigilant on these things than evolved to do right now.
I guess the challenge would be finding those markers that are only for senescent cells and not
Yeah. okay.
cells. That's the same in cancer and all the every
That's
other
right,
field.
that's right.
Same. What those markers are.
So another question I have, is there a progression through cellular senescence that matters, or is it at some point the cell is senescent, that's it, kill it? is there a point where you can reverse? Because, you know, as you think about aging, if all the cells in a tissue are senescent and you kill them, then you don't have that tissue function, right?
That's right.
If they're not, so you still have to have regeneration. So is there, you know, in synthetic biology, we think a lot about kind of like the time of response and being able to look at different temporal stages of progression of whatever, you know, metabolic pathway, whatever it is. Can you do that with aging? Is there a stage in aging where you can still, you know, reverse it, or, and you don't have to kill it, but the cell can, you know, not age and maybe replicate, regenerate?
I think we can do it in at single cell level, of course, like cell aging, we track the aging dynamics all the time, and we can implement our activation or reversal any moment.
But can you, so that, I think the distinction, and maybe you would know the best, is being protective, that is to delay the onset.
That's right.
versus
That's different.
reversible.
Exactly. That's
Yeah.
completely
And so-
different.
And I don't know the reversibility. I don't know that anybody's really talked about that, but does that happen in yeast?
In yeast, yeah, rejuvenation,
You
right?
can make them.
You
young
can make
again.
them young again
Yeah.
by expressing some factors. But I think the most impactful work is done actually in human, in mammalian cell, in mice, like David Sinclair, all these people overexpress this transcription factor.
Yeah, the pluripotent.
Yeah, factors, overexpress 3 of them, and they claim they reverse the whole aging process into semi-stem cell condition, something like that, recover the vision of mice,
Yeah.
old mice.
So it's to restore the epigenetic state. So they're the progression. And this is the kind of thing you would expect to be more reversible than a lot of other damages.
Mhm.
So there, the theory is that the aging is driven by epigenetic decay.
Mhm.
And so if you upregulate these epigenetic factors that then restore the epigenetic markers, then you can recover kind of the normal thing. The part that I don't know, I don't think I know, is how do you know which markers are the original? You need a template for what normal is. And so if the way that you do that is just by upregulating the thing, so you get maintenance, then that's still not reversal. It's
That's right.
It's just maintenance. But I think it's a good point. It's what does reversal look like? So for instance, again, for proteostasis, if you have clots of proteins, are there clot busters that are maybe not? And that's partly what the Alzheimer's folks are looking for, are things that will break up the accumulated plaques. And it may be that you could reverse the physiological state of the cell if you could break up the plaque a little bit, and then the proteasome could start chewing up stuff. So if we knew what was blocking kind of the normal decay cycle, and again, biochemists may know this, I just don't, then you could engineer a drug, or you could do something that would try and get in the way so that that didn't happen. And that, in theory, would be reversible, I think, if you could get rid of the things that are leading to the clogging of the cell.
So the just to make sure I have the terminology right, the difference is between protection and reversal. So when
That's
you
what
use
I think so.
protection,
Yeah.
you just kind of,
You slow down the
Slow-
accumulation.
Slow down the rate, yeah.
reversal would be to actually.
Remove the damage.
Fix it,
You
yeah.
have a lot of damage. So you
I
fix
think
it.
it would be interesting. I think that's a interesting distinction to bring to the broader group, is that,
Okay,
do we think that that's a meaningful distinction? What does it look like to do one versus the other? Does that lead to, is that like a major class of differentiation of research and approach, or is that, are they all the same? I think it's a really interesting idea and point. That
That's
I
right.
don't know this more broadly, maybe it has.
Yeah.
so is this a meaningful distinction, and can we then address it therapeutically?
Yeah.
Or is it worth?
Well, it seems like
therapeutically?
we, so far we've talked about 3 states, right? Protection, reversal, elimination, or something like that. So those are 3 outcomes that could all be positive depending on the tissue. Because if it's irreversible damage, and you haven't been able to protect, then is getting rid of that cell then the best possible outcome.
And so then going back to the idea of identifying markers, if we could then identify markers of this progression of the aging progression, then you could start thinking about addressing either protection, reversal, elimination.
Or you could have like one cell that could do all of those. Depending on like what they sense, they can.
Oh, yeah.
Address all the different things. Would be like the ideal like cell therapy.
Biologist thing.
Well, you kind of need
Yeah.
those markers, because
Yeah.
what does reversible even look like? Like what have you reversed? We don't know. Unless you're just looking at the primary outcome.
Yeah.
You know, the mice can see that, or you don't die, or you don't get cancer. So again, this is part of the fundamental problem with aging, is you have to study old things. It seems to, it seems kind of flippant, but it's really a problem. You can't do things very quickly. Even mice will live for several years. And that's why if we could have human cell or micro tissues that had the appropriate markers so that they look like they were aging, so that, and then we could see what their progression was, and they were a meaningful representation so that we could do things at scale and speed, then I think we could systematically start addressing things. But otherwise, we're just going to have to get what old mice do versus young mice and all that sort of stuff. It's very inferential and fishing oriented these days, I would say. The fact that we still don't know what the cause of Alzheimer's is, for instance, it's really a problem. But it's a problem because it's a human disease in very old people in a tissue that we can't biopsy. So it's really, that's a big problem. And lots of people get Alzheimer's now. Over the next 30 years, it's going to be a huge fraction of the population. It's going to be huge. That's in 10 to 15%. It's going to be really bad.
So is there a good cellular aging model, like mammalian cellular aging model existing?
That's what I don't know, and that's just
right.
ignorant. So that's what I'm not in. Having the human cell folks here, but I think pressing them a little bit more on that. Like I don't know. You would be the person who would know. Do people study aging in human cell lines? I just don't. Usually they're trying to do the opposite thing, is make human cell lines immortal, right? So
That's right. That's right. Yeah,
so I don't.
that's a potential problem here. I mean, how to apply all this synthetic biology approach or even systems biology approaches to a tractable system. I just don't know. Cell kinesis, of course, but it's people.
Is it aging? Is it the same?
Exactly right. I mean, it is the same process. So we can culture fibroblast in vitro, of course, but that's the aging process. What is the aging process? That we can study in vitro or in some way that we can see it.
Yeah, because we still don't want to study that human cell line for 90 years.
No.
So it needs to be somehow inherent or have enough heterogeneity that some fraction of a large cell culture. It seems reasonable in a theory of aging that if it's probabilistic, that if you look at like enough things, some subset of any population will be in whatever that aging state is. And so that might be but if you don't have the markers to sort on those or to identify who they are, then you can't really know it. But if you could grow 1 million cells, you assume that some fraction of them are going to be in a state that is what we think is leading to aging in any cell. Because most cells in our body are not very old. So aging is not necessarily the chronological age of a given cell, except for our very old. There.
So to slow human aging, should we just target cell aging? Is that enough, or organ aging, or systematic aging? What do you think? For animal,
I
because
think you have to do all of it. I mean,
all of this.
I don't know, and this is, it just came up, one of the thoughts. You know, if you cure cancer and heart disease, those are pretty big drivers of
That's right.
mortality, right?
That's right. So focus on aging disease, aging.
Yeah. But it turns out, and I looked at this in detail, that'll actually only shift, maximum lifespan by
A little bit, yeah.
So if you really want to drive healthy aging late in life, it's the unhealth. Some of it's driven by cardiovascular disease, but heart attack might be a cause of death, but unhealthy, you're frail late in life, usually not because of cancer or heart disease, or there's other things.
Mhm.
And so how to cure those or address those. But it does seem like there's got to be systemic hormone-based thing. What's the role of hormone regulation and
Cytokine.
maintaining these things. You've
Yeah.
got the whole leptin insulin system is clearly important. That's in humans and all mammals that becomes more complex and baroque. It's a new function.
How much does like lifestyle factors impact this, or is this just a point where you get there and you're just like, no matter how healthy you eat, how much exercise you do, like the cells are gonna go bad anyways?
Yeah, so lifestyle, again, it's the major driver of all these other aging related disease, like diabetes, cancer, all those things. So you can have a big effect, but at some point, it just doesn't matter.
Doesn't matter.
As I was saying with the runners, like you can have someone who's in their 80s that works out, lifts weights every day. They're still going to be an 80-year-old person with very wimpy arms and stuff like that. We kind of just take that for granted, but that's weird.
Yeah,
Like, why can't you work out successfully when you're 80 years old? What's changed about us? I don't get it. So something, and the fact that we don't understand that seems like a big ignorance problem. But that seems so soft, that's got to be solvable. Because 30-year-olds don't have that problem.
yeah.
It's the same organism, the same DNA, the same functions. So we should be able to figure that out, it seems to me.
I mean,
So
even like
something's given up.
50, like 40, 50-year-olds can.
No.
No. Oh, kids still work out.
Yeah,
Yeah.
yeah, that's
Yeah.
what I'm saying. It's like
Yeah.
not that far away.
So I'm 59, I can still do it. And then I'm like, I'm self-monitoring. It's like, when are these push-ups going to be, they're not going to build muscle, they're just going to maintain, and then when am I going to lose muscle and then not be able to do push-ups?
Remember
Uh,
when broken push-ups?
Yeah,
goes
I'm
down?
at that threshold where I can start sort of self-monitoring
Because
that
of-
transition. Well, why is that? I'm still
Exactly.
I'm still doing push-ups, you know, it just,
That's the biomarker.
to me, it's just weird. It's just not, but that's also what makes me optimistic, that I think those have got to be solvable problems. Like, that's a biological phenomenon. We should be able to understand that.
Mm.
So, and people are working on those things for sure. But back to your point, is that a generalized signal that's going cell communication, the hormonal states? I don't think we really know. To me, it's so systemic. It suggests, I doubt that every single muscle cell in your body is deciding, I'm tired out, I'm just not going to build more muscle anymore. That seems very unlikely to me. I think it's much more likely that there's some sort of state of muscleness that's getting some global signal that says, we're not building muscle now, we're going to do something else. And there's a miscommunication or misregulation that is affecting whole tissues at once. Otherwise, you'd kind of have like one fiber that was really small or something like that. That doesn't seem to be what happened. You just kind of get floppy all over.
So basically, the circulation system is the key. The blood is the key.
Yeah.
To regulate it, systematic aging, right?
It seems like it.
Yeah,
But then what are the signals?
that's right.
Where are
Yeah,
they originating?
that's right. That's right.
Brain based, are they? Don't know. Intestinal.
Yeah, yeah. That to me, I think is a big challenge, even for synthetic biology. Like, I mean, when I deal with a cellular function, a single cell function, then it's much easier, at least, I mean, compared to the animal. When you think about all these organs, all this systematic progression of aging, I just don't know how to solve it. Like,
But again, that's what. Can we create microsystems that mimic the, so in my university, people are, for other reasons, building little organoid physiological systems that look at the muscle, bone, cartilage interface. So,
Wow.
and they get differentiated cells. I don't know how many thousands of cells there are in the thing. But that's a thing that you could then say, does that get old?
Yeah.
Can we maintain it? Could we then screen for signals that maintain building the mother? I don't know if they can exercise the thing. But you know, so I think that's gotta be the approach.
Yeah,
Which we're never gonna be able to do it in the whole human.
that's right,
There's too much going.
that's right. Yeah, that will be very powerful.
So you're thinking we'd have to make a organ on a chip or an organoid of each part of the body that's like an aging.
With multiple parts of the body, right?
Ideally, right, it'd be, well, but you can only go so far.
Yeah, I think each part of the body, but with multiple tissue types.
Yeah.
You know, so you want that interface, so. You know, I really want like a little section of the brain, not just neurons, you obviously need the matrix, and then. Can we make 1 million of those and then screen them with 10,000 drugs? And then if we could, what would we look at? Are any of those, you know, we could then engineer in Alzheimer's related proteins, for instance. So that's another part is not just, it's just occurred to me, not just markers, but you could also engineer sensitized tissues that then you try to cure. So you could pre-engineer aging into a synthetically produced tissue. And then you try to cure that rather than waiting for the tissue to age. You
That's
could,
right.
if you knew what the right markers are, you could create a synthetic
Premature.
aged cell.
Yeah,
And then that could be a model that could then be used broadly in the field to try and fix that cell.
that's right. That's right.
Maybe that's a
Yeah,
powerful way of thinking about that. So if you could figure out, you know, what is the knee, old knee, what's the old brain, what's the old heart slice, and then you look at each one of those. Where you go, because almost certainly, well, it'd be interesting if there's then universal things that can fix all of the problems, but probably there's a lot of
That'd be
tissue
nice.
stuff that.
It looks like the recording will be ending in about 40 seconds.
All right, thank you, Jessica.
Would it need like a progeria cell line that ages prematurely?
there is this kind of.
That's the disease where kids age
Yeah.
really
Yeah.
fast,
Right, it's like
right?
a defect in their DNA repair pathway.
Yeah.
I
Yeah.
think progeria, I think, is the nuclear laminin.
Yeah,
problem.
nuclear laminin,
So
yeah.
Right, yeah.
I don't think that's the right thing. It's just, there's so many ways to make something sick. Just by disrupting global function. That's what happened.
Okay. Yeah, then now I think, now I think we're unmuted, right? Yes.
Map you put up.
We're just. Without the person or?
Yeah, sure.
Okay.
Yeah. Well, that was an interesting map, right? Like looking
Yeah.
at the lifespans and how it is really so different between the different countries.
Yeah.